So, before I get started on the little talk, just wanted to, I came here a year ago, and I came here for three reasons. Four reasons. One was the economy in Detroit was kind of helped push Carly, Mike, and I to move in different directions. The other issue was my wife said we only moved to a warm climate. And my children said moved to the Bay Area and to Santa Monica, and they said they'd give me grandchildren, and so they both have delivered as of 2 weeks ago. We now have, and then the reason I am standing on this podium is because I heard about a year and a half ago that Bob Balow was here. And I said, that is the person I want to work with, that I could be his Tonto, or his Laurel to his Hardy, or his Abbott to his Costello, and do a lot of the clinical work as he spends a lot of time in his lab, and he loves mice and cells and people, but he has to spend a lot of time doing those cells and mice, and so it's been a wonderful collaboration. And we actually put the clinic together very quickly. In about 4 to 5 months, we had the clinic up and running here. So it's very exciting. Cedars has been very supportive of our efforts. It's not easy to put a multidisciplinary clinic together, and the institution has been very supportive. And the CMTA has also been very supportive in helping us develop this. So we thank the CMTA and Pat and Elizabeth. So on that note, I'm going to talk a little bit about substantive issues. So the program said I'm going to talk about the clinical aspect, but I thought what I'm going to do is not tell you about your feet and the things going on with you, because you kind of know that already. What I'm going to talk to you about is, why are you weak? What is actually making you weak? And I hope this is stuff that is helpful to you to understand what's going on with you as you have your clinical picture. So the take-home message here is that no matter what type of CMT you have, the cause of your weakness is the degeneration of your axons. The axons are, thinking of your nerves as wires. The axon is your wire. The myelin is your insulation. And we'll talk more about that. So the ultimate goal is really to stop the loss of axons. No matter which of the 73 genes are affected in your situation, we really want to stop those axons from degenerating and dying. So a brief clinical overview, most of you know this. CMT is relatively common, although it's still in the rare disease category. About 1 in 2,500 people births will have a CMT genetic mutation. That places at least 5,000 people in the LA area, and probably significantly more than that with that. As Pat said, we have 73 genes identified. And just to give you a context of that, when I wrote my first paper on CMT and started working on CMT, we knew of no genes. That was, let's say, 1980. When I moved to Wayne State in 1993, we knew of maybe 3 or 4 genes. When we, about 5 years ago, I'd give this talk, we talked about 40 genes. We're now up to 73, and the explosion of gene discovery in the last 5 years and to date is really remarkable. And Tara will talk to you a little bit about why we've been able to discover all those genes. A lot of it has to do with the Human Genome Project. We can now look at the whole genome and figure these things out much better than we could before. So CMT, my wife Lynn will talk a little bit about the psychological issues, because CMT impacts the lives. You know this, I don't have to tell you this. Impacts your life, impacts the lives of your family, and some people don't always recognize it. The people around you don't always recognize it, and that has impact on everyone in terms of this. So, what do you all have in common with this disease? And I can't say it's 100%, but most of you. Most of you have some variation on a foot like this, Many of you have hands that have some degree of atrophy. Many of you need braces. Some of you have them. A few of you wear them. And one of our jobs is to help you get through the process of dealing with some of these adaptive aids that are helpful to you in ways, but we understand the issues of how it's hard sometimes to accept the fact that you need them. And for children, it's to wear them and deal with their peers on this. It's not an easy issue. So these are the things that everyone has in common. But there's a lot of stuff that's variable. Some of the variability is between the genes defects. Some of them are between families with the genes. So one person with CMT1A, one family with CMT1A may not look like another family with CMT1A. But even within families, there are differences. And that has to do with when you notice it, how severe it is. There are family members, some who unfortunately are in wheelchairs and unable to ambulate, and then there are others who do fine within the same family. Even twin studies have shown twins don't always behave exactly the same. So what that is and why that is always an issue in terms of our understanding of the disease. There are clearly other factors than just those mutations that affect you. So I want to talk about what causes your neuropathy. So you all know that nerves are the things that connect your central nervous system, your brain and spinal cord, to the rest of your body. The sensory fibers send the signals from your body to your brain, and the motor fibers send the signals from your brain to your muscles. We do these things almost instantaneously. So to have that happen, our body has been constructed so that we can send those signals incredibly fast. And to do that, we have nerves. So we have, this is a nerve trunk, and within it there's nerve fascicles, and within the nerve fascicles there are all these little tiny nerve fibers, and those nerve fibers have an axon, the wire, and it has the insulation, the myelin, and the black, and this is a cross-section that's basically one of these, and so the black is your insulation, the myelin, and the white in the middle is the axon. And then you have other tissue that supports that, structures within it. And you can see one fiber diagram where you can see the myelin in the light gray and the reddish axon in between, and you can see these little dimples or indentations here, which are the nodes of Ranvier. So we were constructed to have very thin wires conduct things very quickly. And the way we do that is what's called saltatory conduction, and basically your action potential, the electrical signal, jumps from node to node to node because of these insulation, the myelin. If we didn't have the myelin, to get a fast signal, we'd need to have nerve fibers about the size of your finger to go that, and we would kind of be giants to have all those nerve fibers. So we were intelligently designed so that we can have tiny nerve fibers but still conduct incredibly quickly. So here's an actual single nerve fiber teased from a biopsy, and you can see these nodes of Ranvier. The black is the myelin, and you can see here a little bit of the wire in between. So what does saltatory conduction look like? This is an actual recording from a single nerve fiber done in the 1970s. It's not a computer modulation, but it's basically showing each one of these white dots is a node of Ranvier, and it's a cartoon of it, but this is the actual conductance, and you can see a downward, that's the action potential jumping from one to the other. It's not actually a jump, but it looks like it's jumping. And that is the way the signal goes from in each individual nerve fiber. So that's what happens normally, but what happens if you have a disease of the nerve? So you can do a few different things. You can damage the myelin, and these little oval black myelinated internodes, they're called, can be destroyed. Sometimes they're destroyed just at the edge, and sometimes you lose them altogether. And when that happens, I'll show you what happens to your nerve signal. You can remyelinate. So inflammatory disease like Guillain-Barré syndrome, people become completely paralyzed because they lose their myelin. But many people get almost 100% better because they can remyelinate those nerves. But those myelinated nerve segments are not exactly the same as before. They're a little bit smaller, so they get to be smaller segments. But conduction is restored when the myelin comes back. The other thing that can happen, though, is that you can just damage the wire. Now, this is one of Wallerian degeneration, as if I took a knife and cut the nerve. The axonal degeneration that occurs in CMT starts in the distal end of the nerve and creeps up the nerve. And the longest nerves are the ones that get affected the first. The longest nerve fibers in your body are the ones to the tips of your toes. And then your ones to your arms are not as long as the ones to your feet, which is why usually the feet are affected first and foremost, and the hands become a little bit later. So when that happens, if you have demyelination, you can lose the myelin and have no myelin around it, and then you can get remyelination where the myelin is thinner than you can. This one is probably almost normal, but you can see how this one's a nice large axon, but it's very thinly myelinated compared to this. And the ratio of myelin to axon is the one, if it's perfect, that gives you your fastest conduction. If you have too much axon for amount of myelin, it's slow. If you have too much myelin for the amount of axon, it's slow. So we're made to have just the right, what's called the G ratio. Here's a nerve, a fascicle, where there are very few myelinated nerve fibers left. Here's one here, one here, and maybe there's another one around here, but most of this is, the axons have been lost. This nerve doesn't have any axons working. If you don't have an axon, you don't get a message to your muscle. The muscle is not going to work. This is the T's nerve fibers with some different pathology. So this may be a relatively normal segment except around the edge. This one has these little sausage-shaped things. This is what you see in hereditary neuropathy with liability pressure palsies, kind of the inverse genetic disorder to CMT1A. You can see this in CMT1A, but this is the hallmark of HMPP. You can see some thinly myelinated nerves. You can see a demyelinated nerve segment here. And this area is where the whole axon is just degenerating. So what happens when you mess up the nerve? Well, this is a relatively normal segment. These two little white dots mean that the paranodal region has been retracted a little bit. This is a whole demyelinated segment. This is a shorter internode, one of the remyelinated segments. So let's see what happens when you perturb it. So you get a normal one there, and that one took a little bit longer, and now you have a demyelinated segment. And this isn't actually what would happen pathologically, because basically the conduction would stop. But this is what would happen if you had a normal unmyelinated nerve fiber. See how slow it took to get that little segment rather than jumping? So these are different ways perturbing the myelin will affect conduction. If you mess up the axon, you destroy it, nothing happens, because there's nothing to conduct to. So CMT, here are some of the genes that we see in the myelinated genes, and some of them are in these segments between the rhapsomyelin, some of them are in the myelin. PMP22 is in the myelin, MPZ is the major protein in the myelin, the connexin 32 is in the Schmidt-Lonneman incisors and paranodal regions, and there are some genes that affect just the axon. The difference between your nerves and a wire is that wires are inanimate, and until you stimulate them with putting them in the electric socket, they don't do anything. But nerves are living structures. They're cells, and there are things happening in your axons all the time. Nutrients are going up and down the nerve fiber. The myelin is affecting the axon. The axon's affecting the myelin. So there's a lot of interaction going on here. And one of the things the STAR group is doing is trying to understand this interaction, because understanding it is what's going to lead to treatment. So what do we do clinically that has anything to do with this? So that's where my job of giving you as much pain as I can with these nerve conduction studies. A guy has to have some fun. When we give you nerve conductions, in CMT in particular, the first thing we want to know is, do you have the slow conductions or the faster conductions? Slow means myelin problems, fast means non-myelin problems, loss of axons. And as you know, you either have CMT1, CMT2, or the one in between. So this is from our study back in the 1980s, when here's a normal nerve conduction of 56 meters per second. Figure is, us Americans, a yard per second. And here's a CMT patient. We didn't know the genes at that time, but clearly it's 1A. The conduction here is 18 yards per second, okay? About a third of what the one is from the normal. And this is with an inflammatory neuropathy where the conduction was in between, but it didn't look exactly as nicely as this one. So one of the things we noticed was that in inherited neuropathies, the conduction is abnormal everywhere. So what really causes the weakness? Is it that slowing of conduction, or is it the axonal loss? And I told you the answer at the beginning, and it's the axonal loss. So in CMT1A, which turns out to be probably the only form of CMT where there's slowing in every segment almost the same, if I did the nerve conduction to your facial muscle, it would be very, very slow, just as slow as the one to your hand or your feet. And yet most of you, I would say, maybe all of you would say, my face is fine. I smile good. I puff up my cheeks. I frown. There's nothing wrong with the strength of your face, and yet the conduction velocity is 20 meters per second. What causes the weakness is the fact that the axons to your feet and the axons to your hands are degenerating, where the ones which are shorter are not degenerating yet. So slowing of conduction has very little effect on your strength. It's primarily the loss of axons. Now, if you block the conduction, which some disorders do, that's another way you can get weak. But mostly in CMT, it's the loss of the axons. So Tara's going to go over this a little bit. As you know, we define our disorders and we figure them out now that we have 70 by whether you have CMT1, CMT2, whether you have CMT intermediate, whether you have purely sensory or purely motor disorders. So we'll go over this. I'm going to skip this a little bit. So you know that right now we don't have curative treatments for the diseases. We're working on it. There's some hope. There's some good reason to be hopeful for the future, but we don't have them. So really what we do in the clinic is to work with you to try to do the best we all can do to make your life as fulfilling, as meaningful, as active as it can be. But there is a lot of hope, and I just want to spend the last minutes that I have talking about our involvement and the Stanford Clinic's involvement in the Rare Disease Clinical Research Consortium that was developed about 5 years ago by Mike Shai. Carly was very involved in that creation when we were all at Wayne State. We're proud to be the 15th center worldwide in this. Stanford was, I'm not sure what their number was, but they preceded us. One of the reasons I came to California was it was very clear to me there was a very clear need in the LA area and in California in general in terms of dealing with peripheral nerve diseases and CMT in general. You should feel very good that you now have 2 excellent centers in both the Bay Area and in LA to deal with some of these issues. So there are 15 centers. It's NIH funded. CMTA also supports it. We have a renewal in as we speak, and hopefully that will be approved. The consortium has been very successful over the last 5 years. We've recruited over 4,000 patients into our registries from these sites. There's been over 100 manuscripts published from the consortium members related to the disease in the last 5 years. And there are a number of research projects going on throughout the consortium and being developed as we speak. Up. I don't know. I used to have a dog bark to tell me about my timer, but so I just wanted to let you know at the last, if you come to the centers, we would like to register you as a active participant in the clinical research centers. But you can also register yourself through a contact registry, there can be, you can go on the website. They're going to ask you a few questions. There have been about 1,600 patients already registered on this site. This is, if you go to the Rare Disease Network, they will be able to lead you through the process of registering. They are looking for basically patients who have known disorders, but they also will take patients who have unknown disorders. So that's kind of what the website looks like. I will let you know that so far, these are some of the genes that the Rare Disease Consortium has found in the last 5 years. Pretty impressive, isn't it? They also have a training program. There have been a number of people who have been trained, fully supported by the RDCRN, to work on CMT. And almost all of these people who are staying as they develop their careers doing work in CMT. Otherwise, how are we else going to get young people to work on our disease? So on that note, I thank you very much. And I was almost on time. So Glenn, let me get you up here. While we're getting Glenn, he's switching computers on us.
Starting 10, right?
Huh? Am I early?
You got 20 minutes.
Oh, how did I get so
Or
early?
15. What time do you have? You have till 10 o'clock.
Okay.
So
So I have a few minutes to answer any questions. Yes.
Does anybody ever have CMT from more than one gene?
It's possible. There are probably
The question is, does anybody have CMT from more than one gene?
Carly had it.
So,
Should just go right.
based on a study we did with the Wayne State Group in 2011, we had 11 families out of 600 that had 2 forms of CMT that were identified. So it was about 1% or a little bit under 1%.
Sir. Sir. It's not set.
As Tara will go over in a little bit, the genetics has become better, cheaper, but more
Yeah, it doesn't
complicated.
show. They're all missing.
So everyone has mutated genes,
Sometimes it is.
all of us. Most of them don't do anything to us, but a few do. And sometimes the trick is, are we sure that the genes that are found are really causing the disease?
Yeah.
These 11 families clearly had 2 causative genes. There were not 2. One was what we call a polymorphism or a benign mutation.
Here, let's hear that. Oh, you know what to do.
Hi, good morning. Perhaps you covered it already, but I got here late, and I apologize if you have already. But type 2B with gross mutations, is there any research going on related to CMT type 2B?
There's a fair bit of research going on 2B. That's the disorder. Well, in 2B, I don't know if there's a ton of. I'd have to look into it. The problem we have, when you have 73 genes, that may be more genes than we have labs doing the research. So what I will tell you, almost every gene that's been discovered, there are people looking into the mechanism of what that gene does. So when I recruited Mike Scheid to Wayne State 20 years ago, or 19 years ago, Mike said, I want to work in CMT, that's all I want to work on. And he said, because the way we're going to understand peripheral nerve function and peripheral nerve disease is to find genes and find out what they do, and then find out what disturbing them does. So there are people working on each of the genes. There are labs always looking to try to figure out the function and what happens to those mutations. But clinically, I can't say that specifically 2B is being looked into.
What is the cost to register at the Cedars CMT clinic?
I'm going to will defer to Tammy on this to some extent. The Cedars Clinic is not free. We do bill insurance. I don't control the billing. We've worked out some mechanisms with Cedars for those who don't have insurance that will pay to minimize or limit the cost. But until we have research funding that would allow us to see people for no cost, we work through the insurance. So what we do is if you call and say, I would like to be seen, Tammy works this out with you. She either says, your insurance is great, we don't have any problems. Your insurance is lousy, and...
I rarely say that.
She's nicer than I am, but...
I'd like to say it more.
But we try to work it out with you. And if there's a research issue, then we will sometimes see you for research, which would basically be no cost. And if your insurance will cover seeing Dr. Balo, but not the physical therapist, we work that out. So we try to do whatever we can to help you. I mean, we do the best we can on that.
Yeah, I was just going to comment that the research components, giving your DNA for research, doing testing for the neuropathy score for the Rare Diseases Network, all those things are not charged for. It's really just the regular visit type things. So if we can get you in, we can certainly usually do those things, and that's great.
We have one question over here.
I heard Dr. Shai speak a few years back, and he said that CMT is symmetrical. Myself, my entire family, and there's tons of people that I've met that CMT is not symmetrical. So what research is being done in terms of that, and why am I still hearing that it's symmetrical when it very clearly is not for a lot of us?
Do you know the genetics of your 1A?
I very clearly can tell you my right side is severely worse than my left side. It's had 8 surgeries, whereas my left, the very first surgery, was good.
The short answer for that is that just like I mentioned, there are things that we all have the same and things that are variable. The symmetry is not always so symmetric. So I think, not speaking for Dr. Shai, but it's not that one side doesn't have it and the other side does. The severity can be variable, and we don't know the reasons why. We know that every Schwann cell in your body has the PMP22 duplication. But why it's expressed more or less in one or the other, that's an area that is being researched in terms of understanding gene expression, modifying factors, and other things of that sort. Did I do good, Bob?
I'll throw out there that I sometimes show pictures of Charcot's first paper in 1869, right? Actually, no, that one was 1880-something, sorry. Mm-hmm. And in fact, there was one patient who was extremely asymmetric, even from the first descriptions. So you'll see that one calf and leg was much more atrophied than the other. So it's pretty well known that that happens, but I would agree with everything else that Rich said as to why, we don't know. So.
Hasn't there been work of modifying genes or modified genes that have explained
Right.
versus
We won't jump ahead too much, but absolutely. So we don't know why. There are 2 possibilities. It's genes and it's environment. And the genetics, we are actually a bit better at figuring out. So there are projects within some of the people who have 1A, for example, their DNA is going to Miami, and we say, well, we already know which type they have. But actually what we can do with that form, since there's so many patients, is say, okay, well, why is it asymmetric in these patients? Why is it more severe in these patients? And can we use that insight to develop treatments? So that's a really good point.